Thank you, Chairperson. I'm Hiroshi Shimura of Yamanashi University in Japan. The title of my presentation is Therapeutic Potential of Cell-Type Selective Optogenetics for a Mouse Model with Urinary Frequency. My COI is as follows. The technology of optogenetics is useful for understanding the mechanism and developing novel treatments of neurological disorders. Optogenetics needs an expression of light-activated protein opsins for target cells, and light illumination for the cells enables to switch on or off the action potentials of the cells. Optogenetics has a large advantage of cell type specificity and temporal specificity. Cerebral cortical neurons consist of excitatory neurons and some inhibitory neurons. Optogenetic approach enables to focus on the specific type neuron. There is no evidence of optogenetic approach targeting central nervous system. We already have shown that the anterior cingulate cortex ACC regulates urination, activating excitatory neurons and interneurons induce and suppress urination respectively. In the present study, we evaluated the effect of autostimulation for interneurons in the ACC using urinary frequency model mice. In preparation for the light stimulation in the present study, we need virus injection into the ACC of transgenic mice. Adeno-associated virus DIO channel rhodopsin injection for PV mice could result in an expression of excitatory opsins channel rhodopsin only for PV neurons in the ACC. PV neuron is one of inhibitory interneurons. After 1 month, bilateral optic fiber implantation and cystostomy were performed under urethral anesthesia. In cystometry recording, the perfusion was normal saline for control or acetic acid for urinary frequency model. After stabilization, photostimulation was conducted and intercontraction interval ICI was measured. In order to minimize the variation of ICIs, we collected 5 ICIs under each condition and took each average of those measurements. We normalized the ICI before manipulation to minimize differences of ICI baseline between individuals. Statistical analysis is as follows. This is the results. First, photostimulation for channel rhodopsin expressing at the ACC PV neuron mice prolonged ICI during stimulation phase only, and ICI was recovered after photostimulation with indicating temporal specificity. Otherwise, photostimulation for wild-type mice demonstrated no significant difference between any phases. Next, overstimulation greatly prolonged ICI, but ICI didn't recover even after stimulation. Bladder tension test in organ bath study shows that stimulated bladder had a less contractility than moderate stimulated. This outcome would implicate that overshooting bladder capacity causes bladder distension and bladder tissue damage. Finally, we confirmed that acetic acid perfusion makes urinary frequency model, and even in acetic acid cystostomy, photostimulation prolonged ICI during stimulation phase, and ICI was recovered after photostimulation. In acetic acid model, the ACC would receive signals from activated afferent nerves and regulate micturition to void frequently. In the present study, PV neuron activation in the ACC would have suppressed the frequent voiding signal. PV neuron selective photoactivation in the ACC succeeded in prolonging ICI even in a disease model. Applying optogenetics to treatment for lower urinary tract dysfunction have an advantage of temporal specificity, which would be useful for controlling immediate agency. Too strong stimulation would cause concern of bladder distension. Changeability of stimulation power could avoid an adverse event of bladder distension. This is a first study applying optogenetics technology for treating urinary frequency targeting central nervous system. The advantages of cell type specificity and temporal specificity could be epoch-making for treating refractory lower urinary tract dysfunction in the future. Thank you for your attention.